The process of evaluating the biocompatibility of materials will be reviewed in 5, including the present, lectures. The title of Lecture 25 is Evaluation of Biocompatibility, In Vitro, Part 1, Material and Mechanical Tests. Before we start the review of the various stages and tests which are required in order to establish the biocompatibility of materials, a reminder regarding the prerequisite tests which must be completed successfully before any evaluation of biocompatibility is attempted, namely that the materials and pertinent devices must be characterized in vitro regarding their functional performance, and for that purpose, prototypes of the various devices are used, and those tests must be successfully completed. In vitro mechanical type tests are used in this process. The principle for the mechanical test is that during this testing, the material sample or specimen is subjected to uniaxial loading at a controlled amplitude and rate either once or repeatedly. Of course, in the case of repeated testing, the appropriate test is the ones related to determining the fatigue results. The types of mechanical tests used are tensile, compressive, bending tests, creep tests, stress relaxation, torsion, and hardness related tests. As a result of these tests, data are collected and they can be plotted in stress versus strain plots or stress strain versus time plots. Based on these particular plots and data, calculated parameters which are pertinent to mechanical characteristics of the materials are the moduli, yield strength, tensile strength, toughness of the material, and conditions of failure of the specific materials under testing. Characteristics and comments regarding mechanical tests include the fact that these particular tests are destructive usually to the specimens of the material which is tested. They require, of course, availability of special instruments and the use of pertinent protocols. They are needed, however, in order to determine the suitability of materials for biomedical applications, especially applications which will expose the material and the device under mechanical loading conditions. The pertinent prerequisite tests also include in vitro material-related tests. A very important aspect which must be carefully considered and tested is that of the degradation potential of biomaterials in the physiological environment. Uncontrolled degradation, especially following implantation, is undesirable and should be avoided. However, in some cases, controlled degradation of a material is incorporated in the design for a specific purpose. Such biomaterials, biodegradable materials, are used in tissue engineering applications in the cases of controlled release of drugs, etc. This is a major difference. If that particular aspect is incorporated in the design for a specific purpose, it is allowed and should be incorporated, but also it has to be appropriately tested in order to assure that the purpose of the tissue engineering scaffold, for example, has been met, or the purpose of the controlled release of certain medications and drugs is also met. Quantification of biomaterial degradation follows a well-established protocol and procedure. It is an in vitro testing. The material samples of certain specific dimensions and weight are placed in vials or containers, which also have a simulated body fluid. For example, one that has the chemical composition which is similar to that in vivo, certain specific pH, etc. And these particular now specimens immersed in that simulated body fluid are maintained at 37 degrees centigrade for various periods of time. At prescribed time points, the following tests are performed. First, aliquots, which means small amount of the supernatant bathing fluid, are taken and analyzed for the presence and composition of any components which are eluted from the specimens of the tested materials. For example, ions, if they are metals, and monomers, if they are polymers. Each material sample is inspected visually at the end of each time point for signs of degradation. For example, did the color of the specimen change? The shape change? The appearance of cracks has occurred compared to the controls, which of course are maintained under conditions which are pertinent for that purpose. Another important aspect, and that's why we start with materials which have been weighted and that particular datum has been recorded. Mass loss of each material specimen is determined at each prescribed time point. At the end of the experiment, certain physical and chemical material properties are measured and compared to the values of the respective properties of the same sample before in vitro testing. In all of these cases, the important aspect which we must keep in mind is the fact that we need to have appropriate controls for all these tests and at all stages of the evaluation process. In the case of implantable metals, the process of degradation is known as corrosion. This results from oxidative reductive chemical reactions which take place in the physiological environment or milieu. The process sometimes is affected if the handling of the metal specimen is not done carefully, and if it causes, for example, cracks and crevices and pits. This particular topographical change may promote and enhance the subsequent process of corrosion. Corrosion of metals can also result from the removal of the passivating layer on the metal surface by either mechanical or chemical means. What we mean by the passivating layer is the following. Remember that any material which is introduced inside the body within seconds is covered by proteins which are abundant in body fluids and in the body tissues. Sometimes specific metals promote the interaction of certain proteins which, once the metal surface is covered by them, usually that protein is albumin, the oxidative reduction chemical reactions, the specimens which are produced like oxygen radicals, cannot reach the surface of the metal and therefore start the process of corrosion. This passivating layer, therefore, results as a protective layer against corrosion of metals. Degradation of metals also is affected by the biological environment. proteins, lipids, cells, bacteria, and all the chemical compounds that the activated cells and bacteria may release can be attached on the surfaces of metal implants, and they may start the process of corrosion. Ceramics are materials which are used for several biomedical applications. These materials, of course, are not recommended to be used in applications under dynamic loading conditions. For example, they cannot withstand compression. However, these materials are desirable because they promote interaction of bone cells. We must keep in mind that the inorganic phase of bone is a ceramic, specifically hydroxylapatite, and bone cells interact very well with that particular environment in physiology, and of course they interact very well with ceramics, which biomaterials, biomedical engineers produce and provide for applications in bone. A particular example of such an application is when, let's say, metallic implants like a hip prosthesis, the surface of the metal is covered by a ceramic. That particular now composite implant has the mechanical strength of the metal to withstand the mechanical load of the hip, but at the same time, because of the ceramic coating, provides a very attractive surface on which the cells from the surrounding bone will interact and start forming new bone. Ceramics in the physiological environment degrade primarily as a result of infiltration of water at the pH environment of the body. They are susceptible to failure via stress-induced degradation, especially in an environment which has mechanical dynamic conditions. The rate of the degradation of ceramics is controlled by the susceptibility of the specific material, because there are different ceramics. For example, hydroxylapatite, alumina, and titania. Notice that the end of those words is an A, not aluminum or titanium. Alumina and titania are the ceramic layers formed on the metals aluminum and titanium respectively when those metals are exposed to an environment which has oxygen, like air. Another parameter for the rate of degradation of ceramics is the degree of their crystallinity, and last but not least, the amount of water available in their environment. Many implantable devices these days are composed of polymer materials. Degradation of polymers takes place via swelling because water infiltrates their structure, and in the water environment and the pH of the physiological milieu, they start swelling, and slowly but for sure that particular process can cause their degradation. They break down because the polymer backbone chain of the carbon atoms breaks down. This breakdown can occur through hydrolysis. This is the official term which describes the presence of water and the infiltration of water into the polymer environment, which slowly but for sure causes the swelling and then the breakdown. Another way by which the polymers can break down, the backbone chains can break down, is oxidation. And this, of course, is caused by the presence of reactive free radical species such as oxygen radicals. Last but not least, the enzymatic reaction of the chemicals present in the physiological environment as a result of the various processes which we already reviewed, like blood clotting, activation of white blood cells, and other events at the tissue level. All these chemical bioactive compounds, special enzymes, can bring about the degradation of polymer compounds. Because of the wide use of polymers in biomedical applications, they have been studied extensively. One aspect which has also been studied extensively is their degradation. There are two types of hydrolytic degradation of polymers. One of them is referred to as bulk hydrolysis. This refers to the invasion of the water into the bulk polymer at such a rate that causes the polymer breakdown. As a result of this particular breakdown, there are many water-soluble degradation products. Some of them will be the monomers, for example, of the specific polymer, which is degraded. Another type of degradation of polymer is what it's called surface hydrolysis. This process causes the degradation of the polymer as the surface is exposed to the water environment of the body and gradually, layer by layer, moves towards the center of the specific polymer specimen. Examples which are not related to the biomedical applications but may help us visualize these two processes of degradation of polymers in a water environment are the following. In the case of the surface hydrolysis, it will be the situation which all of us have experienced, namely the way a bar of soap slowly but for sure, layer by layer, is exposed to water and it's dissolved, and slowly but for sure that particular dissolution makes that particular bar of soap smaller and smaller and thinner. An example of bulk hydrolysis will be the case of a cube of sugar. If you are drinking tea, for example, because if you put it in coffee, you're not going to be able to visualize this process. If you drop a cube of sugar in your cup of tea, then you notice that that particular little square stays intact for quite a bit of time, but all of a sudden, and this is now at the time when a lot of water has invaded its three-dimensional structure of the ceramic, which that's what it is, that sugar cube, it collapses. And that is the evidence of bulk hydrolysis. In closing this lecture, a reminder again of the prerequisite test for the evaluation of biocompatibility. In vitro characterization is needed before evaluation of the biocompatibility of any material may proceed. Because if the material in the device does not pass this prerequisite in vitro characterization test, then that particular material and device should not be evaluated for their biocompatibility. The process stops there, and biomaterial scientists and engineers have to go back to the drawing board and start the process of design, formulation, and characterization of the novel biomaterials from the beginning. This is the end of Lecture 25, entitled Evaluation of Biocompatibility, In Vitro Part 1, Material and Mechanical Tests.